Hey everyone, welcome to this spontaneous and not quite Hollywood budget csRNA-seq methods tutorial. In this video we'll run you through how to perform the method. For more details about the method, please check out our manuscript. But we'll also hope to add an additional video in the near future that discusses the method in more detail. For now it should be suffice to say that the cool aspect of csRNA-seq is that it can capture ongoing transcription of both stable and unstable transcripts, such as enhancer RNAs, from total RNA. The method is thus readily applicable to any fresh, frozen, and even fixed tissues, clinical samples, and also helps the analysis of pathogens or viral samples under BSL-2 conditions. More infos about the method can also be found on our HOMA website or the contact details below. But for now, let's get started. Pre-run your commercial or homemade 15% TBE urea gels for 20 minutes at 200 volts. Meanwhile, while the gel is running, you can mix your RNA. Add anywhere between 3 and 25 micrograms of total RNA in a volume of up to 14 microliters with an equal amount of formaldehyde loading buffer. Note that we were able to successfully perform the assay from inputs as little as 300 nanograms. However, that is not recommended for the beginning. Next, heat denature your RNA at 75 degrees for 2 minutes if you use PCR strips and 2.5 minutes when using 1.5 milliliter tubes. Quick chill your RNA on ice. Then blow out your gels well to remove any excess urea and carefully load your RNA with generous spacing. As shown here, I like to use these filter gel loading tips that you will see a number of times throughout the tutorial. Run the gels for 45 minutes at 200 volts and subsequently stain them using gel green or ethidium bromide. Important, it is not recommended to use high sensitive dyes such as cyber gold, as these are more likely to light up your small RNA. To size select our short RNA, we can now cut just under the first steady state RNA band, which is nicely stained by the ethidium bromide. The exact location of this cut varies from species to species, but it's usually between 59 and 61 nucleotides and right underneath the xylene cyanol. On the lower side, we cut about 2/3 down between the two dyes. As seen in this picture, this is a sort of negative selection area where the RNA is barely visible. It is important to note that you do not need to seed anything to successfully perform the method. And indeed, as shown in the well furthest to the right, a prominent smear is more indicative of degraded RNA than of small RNAs. However, if you would load, let's say, 15 micrograms of nuclear RNA, you should clearly see the small RNA band and most strikingly the microRNAs. To more efficiently elute our RNA, we will shredder the cut gel slice. While shredder tubes are commercially available, you can also just take a 0.5 milliliter PCR tube and punctuate it 3 to 4 times with a 20 gauge needle. Next, spin this setup for about 5 minutes at maximum speed. I would recommend to use an Eppendorf centrifuge with low acceleration to not lose the lids of the tubes. The gel should form a nice slurry at the bottom of the 1.5 milliliter tube. Following the shuttering, transfer the slurry into the 1.5 milliliter tubes and add 350 microliter of csRNA-seq elution buffer. If you're new to the method, you may consider adding some RNase inhibitor here too. Ensure the slurry is nicely resuspended in the csRNA-seq elution buffer and elute the RNA under gentle agitation for about 2 hours. While the RNA is eluting, prepare for each sample 1 millipore ultra-free spin column and one labeled 1.5 milliliter Eppendorf tube that contains 1.5 microliters of 15 mg per ml glycoblue. After the elution, concentrate the slurry to the bottom of the tube by spinning it at 500 G for 20 seconds. Transfer the slurry into the spin columns using a wide bore filter tip. Every 5 or 6 transfers, I would recommend to cut off the tops of the column and transfer them into the 1.5 milliliter tubes that contain the glycoblue. Once all samples and spin columns are transferred, spin the samples at 1000 G for 1 minute to separate the gel slurry from the eluted RNA. Discard the spin columns and then add 3 volumes of ethanol, 100%. Close the tube, mix the sample well, and precipitate the RNA in a -80 freezer overnight. The next morning, start by spinning down your RNA for 30 minutes at max speed at 4 degrees. If you do not see a pellet, vortex the tube hard and spin for another 5 minutes. Seeing the blue glycoblue pellet is independent of the amount of RNA, so ensure that you recover a nice blue pellet. Meanwhile, while the RNA is spinning, you can prepare 4 things. First, 1 set of empty PCR strips that are just labeled. These will contain your csRNA-seq samples. 1 set of small RNA-seq samples, strips, for each of which you add 1 microliter of TET. You may also prepare 1 set of PCR lids that contain 6 microliters of TET and a terminator master mix. Once the sample is done spinning, aspirate off the supernatant without perturbing the pellet. Quick spin the tubes and remove the remainder of the ethanol using a filter gel loading tip. Add and wash the RNA pellets in 400 microliters of 75% ethanol. To minimize the loss of material, we next transfer the RNA as a glycoblue pellet rather than resuspending it. To do so, pipette the pellet in 75% ethanol with a P1000 into the PCR strip tube. Next, quick spin the PCR strip and remove all remainder of the 75% ethanol. Dry the pellet for about 3 minutes. Once all pellets are dried, you can now add the PCR strips containing the 6 microliters of TNT to close the tubes. Vortex the samples well till all the RNA samples are well resuspended. Next, heat denaturate the RNA as shown earlier using a PCR machine for 2 minutes at 75 degrees and quickly chill on ice. While cooling down, you can add 14 microliters of terminator master mix into 3 sets of PCR lids. Next, it's important to not forget the input. Therefore, quick spin the PCR tubes, remove the caps and transfer 10% or 0.6 microliters of the small RNA mixture into the input PCR strip tubes that contain the TET. Terminator, or its buddy XRN1, is a 5' monophosphate dependent exonuclease. Therefore, terminator readily degrades 5' monophosphorylated RNAs, including most ribosomal RNAs and microRNAs. To do so, mix the terminator master mix that's in the lid of the PCR strips with the small RNA. Spin down and incubate at 30 degrees for 1 hour. To reduce the overall time of the protocol, I commonly prepare the master mix for the next step and aliquot it into the lids of PCR strip tubes towards the end of the incubation time from the previous step. As shown here, with just 5 minutes left of incubating the terminator reaction, aliquot the SIP1 master mix into the lids on ice. Once the terminator reaction is completed, then you can just quick spin the samples, remove the lids, and add on the lids that contain the master mix for the next reaction, in this case the SIP1 master mix. This is a quick way, and you also save a bunch of pipette tips. Again, mix the samples well by inverting, quick spin and incubate at 37 degrees for 45 minutes. This gives you a nice 35 to 40 minutes to relax or get a coffee until we prepare the beads purification step. For this step, add 50 microliters of AMPXP RNA ampere beads into the lids. Once the reaction is completed, quick spin the samples and place them on ice. Remove the caps and exchange them with the lids that contain the beads. Mix the sample well, quick spin, then remove those lids, but keep them. Add 1 total volume or 100 microliters of 100% isopropanol. Close the sample. Mix again well, quick spin, and incubate on ice for about 10 minutes. Under these conditions, RNAs with the sizes that are larger than about 10 nucleotides precipitate to the beads. Following this 10-minute incubation on ice, collect the beads on a magnet for 3 to 5 minutes. Make sure the liquid is nice and clear and all beads are carefully collected at the magnet. Do not aspirate the liquid as it contains speck. Instead use a pipette. Next, wash the beads twice with 80% ethanol. Prepare this ethanol fresh. A good way to wash the beads is to use magnets to make the beads fly from one side to the other side of the tube. After each wash, collect the beads and aspirate off the ethanol. After the second and final wash, quick spin the sample one more time to collect all the ethanol. Use once again a magnet to collect the beads and aspirate off all remainder of ethanol. Let the beads dry for 3 minutes, but it's important to not extend this time beyond 3 minutes. While the beads are drying, prepare lids that contain per sample 10 microliters of TET. Following the 3 minutes, place the lids with the TET onto the tubes and make sure that the beads nicely resuspend. This may take 3 to 5 minutes. The next step is absolutely critical to further deplete microRNAs. Heat the RNA beads in TET at 75 degrees for 2 minutes and then, as you did before, quickly place them on ice. This repeated denaturation helps to further deplete particularly the microRNAs, as their secondary structure often protects the 5' ends from dephosphorylation or the 5' terminator exonuclease. Next, exchange the lids to add the CYP2 master mix. Mix the samples well, quick spin, and incubate on 37 degrees for about 40 minutes. This gives you another nice long break, in which, however, you can already label 1.5 ml Eppendorf tubes in preparation for the trisol extraction. This extraction is important as beads purification also carry over protein in the next reaction, and we do not want to have any of the 5' enrichment proteins in our library prep. Following the incubation, add 150 microliters of TET to the CYP reaction. This is just done to increase the volume of the reaction. Keep the samples on a magnet and pipette the samples starting from the bottom of the PCR tube into the 1.5 ml tubes. Do not transfer the beads. Once all samples are transferred, move to the fume hood and add your 500 microliters of trisol LS. Please read the SDS for trisol if you're not familiar with it. It's quite nasty stuff. After adding the trisol LS, cap the tubes, vortex well and quick spin. Next, add about 150 microliters of chloroform to trigger the phase separation. Again, vortex the samples extremely well, and then some more, and then spin them at room temperature, at about 12,000 G for 10 minutes. You could also spin them at 4 degrees, however, you may sometimes see the proteins accumulate in the interface. While the samples are spinning, prepare another set of 1.5 ml tubes containing 65 microliters of 3 molar sodium acetate. If you had nice glycoblue pellets, you do not need to add any additional glycoblue here. However, if the pellets were a little weak, this is your chance to make them a little bit bolder. Following the spin, transfer the supernatant of the samples into the new tubes. Take as much as you can, but make sure it is clean supernatant. It is important to first mix the RNA with the salt, then quick spin before adding the isopropanol. Once the isopropanol is added, mix the samples well and incubate it for at least 20 minutes on ice. You can also incubate these samples at -20 overnight, but do not incubate them at -80. Following the precipitation, Spin the samples for 30 minutes at 4 degrees at max speed. Aspirate off the isopropanol, wash the pellets, and transfer them, as already shown in the beginning of the video. To start your library prep, you want to have the RNA as a glycoblue pellet in a PCR strip tube. Good morning! Well, now that we have both 5' enriched RNA that I will refer to as csRNA, as well as the input RNA, or the whole small RNA, we can start with the library prep. If you haven't already done so, resuspend the csRNA-seq pellet. To do so, I would pipette 3 microliters of TET into PCR strip lids, and then simply exchange the lids, as done so many times before. Make sure the RNA is nicely resuspended. Fetch your input RNA from the freezer, and once everything is ready, heat denature the RNA at 75 degrees for 2 minutes, as done previously. Quick chill the samples on ice and start preparing your RPPH master mix. This is an art by itself. While the Crowder PEG is not essential for this reaction, it facilitates adapter ligation downstream in the protocol. However, given the viscosity of PEG, it is now important to, particularly in the first 2 steps, vigorously mix your samples. Start off your RPPH master mix by pipetting the T4RNA ligase buffer into a 1.5 ml tube. Next, add the RNase inhibitor as well as the PEG. Consider using a wide bore tip to pipette the PEG. Mix everything vigorously by inversion and vortexing, then quick spin before adding the RPPH. Once the enzyme is added, again mix the master mix really well, and then pipette 5 microliters of the master mix into PCR strip lids. For input samples, you should consider just using half, or 2.5 microliters. However, if you have problems with the small volumes, you can also increase the volume of input samples to 3 microliters and continue with exactly the same protocol as for csRNA-seq. The smaller volumes are simply because it works and it saves money. Add the RPPH master mix samples onto the RNA tubes. Mix well by extremely vigorously inversion. Yes, I might sound like a broken record, but that is a little bit of a challenge. Mix some more, then quick spin, mix one more time, and then incubate the samples at 37 degrees for an hour. Well, now you have a good 50 minutes to recover before we start preparing the 3' master mix. Here you can either use a home brew setting as described in the Hetzel paper with T4RNA ligase 2, or use kits like the small RNA kit from NAB, which I personally like a lot. For beginners, I would recommend to just start with the NAB small RNA kit. Here, mix 2 microliters of 3' NAB adapter buffer, which already contains PEG and a lot of the nice stuff, with 0.3 microliters of 3' adapter and 1.7 microliters of the enzyme. Here it is critical to know that the amount of adapter concentration relative to the small RNA determines the amount of ligation efficiency as well as of adapter dimers. While we use hybridization to deplete adapter dimers, if you find yourself ending up with too much adapter dimers, reduce the amount of 3' adapters. For example, with patient RNA, where I sometimes have less than 500 nanograms, I use 0.1 microliters, or sometimes even 0.05 microliters of 10 micromolar 3' adapter. Again, after mixing everything nicely, pipette 4 microliters for the csRNA-seq and 2 microliters for the inputs into the lids of PCR strip tubes. Once the RPPH mediated decapping reaction is done, you can now quickly exchange the lids and incubate the samples at 22 degrees for an hour to 2. Note that the RPPH enzyme would also remove the adenyl group from the 3' adapter. However, it is inactive at 22 degrees. Once your 3' adapter ligation is completed, you can add the RT primer. To do so, I recommend diluting the RT primer to about 5 micromolar and then using a multi-channel to add 1 microliters of RT primer for each sample. Hybridize the primer by heating the sample to 75 degrees for 2 minutes, followed by a 30 minutes incubation at 37 degrees and 15 minutes at 25 degrees. Meanwhile, you can prepare the 5' adapter mix and already pipette it out into PCR strip tubes. Once the hybridization is completed, well, guess what? Yeah, you can quickly now exchange the lids, mix the samples and start the reaction. This gives you another 50 to 55 minute break till you prepare the RT master mix that again you can add into the lids, and once the 5' adapter ligation is completed, exchange the lids. You got it. Mix the samples well, quick spin and incubate the RT reaction at 50 degrees for an hour. This should be plenty of time for you to prepare the PCR reaction and the barcodes. As with the other reactions, I pipette the PCR master mix into the PCR lids, and then, usually using a multi-channel, add the barcode primers. Once the RT is completed, well, we just exchange the lids and start the PCR reaction. If this was all a little fast, please note that below we provide a PDF with detailed instructions, or visit our home website for the latest version of the csRNA-seq protocol. Prior to loading the PCR onto the gel, I highly recommend a bead cleanup. This is to remove excess salt, which allows the DNA to run in a sharper pattern on the gel. For csRNA-seq, this size separation helps to accurately avoid both adapter dimers as well as the occasional 51 or 60 long nucleotide small RNA that might have slipped through the protocol. To do the bead cleanup, add 1.5 volumes of 20% PAC, 2.5 molar sodium acetate, including 2 microliters of carboxy beads, to the PCR samples. This is our homebrew protocol, however you can also just purchase them commercially. Mix the samples well and incubate at room temperature for 10 minutes. Quick spin and collect the beads on a magnet for 3 to 5 minutes. Pipette off the supernatant, then wash the beads twice with 80% ethanol and let them dry super well. Excess ethanol will cause that once the loading dye is added, the DNA will run out of your wells, which is annoying. Unlike for the RNA step, at this point it's totally okay to overdry your beads. While the beads are drying, you can already add about 25 microliters or even more of 1x DNA loading buffer to the lid. As a rule of thumb, I only load 50% of my PCR reaction on a gel. This is that in occasions the 10% TBE gels that we purchase are faulty, or God knows what can happen. However, in short, just adding 50% of the PCR reaction should not really make a difference for the final sequencing run. Next, when loading your DNA gel, remember it is important to load input next to the csRNA-seq sample to ensure that when you cut, the size selection is identical for both. In general, it's also good practice to avoid the outer lanes. Run the gels for as long as possible, which may require some optimization. With my gel setup, I run the gels for 15 minutes at 80 volts, followed by 73 minutes at 180. If you're in a rush while the gel is running, prepare gel shredders. This would allow you to continue to the DNA cleanup within an hour. Otherwise, if you're lazy like me, just cut the gels, maybe slice them in half, and then elute them overnight in 150 microliters of gel elution buffer. Whether you had a slurry or eluted overnight. Next, you can purify your libraries using the Simulcate. Add 5 volumes of DNA binding buffer and transfer everything onto the columns. If you had a slurry, after the first wash, flick off the gel pellet. Otherwise, purify DNA exactly as described by the manufacturer. However, I would recommend to elute using sequencing TAT and not the provided elution buffer. I commonly elute csRNA-seq samples in 20 microliters and input samples in 60 microliters of sequencing TAT. For quantification, I would recommend incubate using the high sensitivity DNA kit. We hope that this overview video helps you to successfully perform the method. Note that there is also an FAQ as well as a detailed protocol online, or find it on our HOMA webpage. Thank you and good luck.